Thank you very much. Thank you very much. Thank you very much for the opportunity to present some of these results of our studies. This very interesting Congress, and I would really like to thank the organizers for setting up this very nice conference. And can you see my screen?
Yes.
Okay, great.
Yes.
So this talk will be shared with my colleague Jennifer Morinay, which is already connected. So at some point I will let the floor to her to talk about some parts of this research. The title reflects what we have been doing under Lifefico to actually perform the hacking activity. So these are preparatory. These are the results of preparatory action we have been realizing for performing the hacking activity that Michelangelo has already been talking about. Here is a brief summary of what Lifefico is about. So as Michelangelo said, so I will not repeat what he said. It's about strengthening the northernmost population of the species, which are likely to become very important in the coming years, because climate change and especially global warming is really making the southern part of the range of the species likely unsuitable, climatically unsuitable in the coming years. We are already seeing now, right now, this very clearly, for example, in Matera, where we have following the reproduction, and we see that many nestlings are dying in the nest boxes because of the heat wave that is hitting hard, very hard, Italy in these days. We are having temperatures about 40 degrees and 39 degrees in Matera right now, and this is really bad for the nestlings in the nest boxes. So the idea of Lifefico is really to kind of favor the colonization of northern breeding areas, which the species already started naturally to colonize in the past years in the population, and to foster the population in these areas. This will be in Lifefico. This will be realized also by Action C2, which involves, in fact, the translocation of some nestlings from Matera to northern Italy and to hack them in the population, where they hopefully will come to breed in the coming years using the protocol and the approach developed by DEMA. So the Ambiente de Colonia approach with adults in a cage, which can attract and provide an attraction for lesser kestrels, for young lesser kestrels, and by releasing nestlings, which kind of get accustomed to the new study site, and hopefully will return to breed there. In this hacking activity will be performed in one premiliar or nesting tower of lesser kestrels, for lesser kestrels, specifically produced by Lifefico. But before deciding how to do this action, it was important to perform preliminary activities to identify which was the best source population for translocating these nestlings to northern Italy. In fact, the International Union for Conservation of Nature provides very strict guidelines for reintroduction or translocation of animals for conservation purposes. And the life regulation requires life proponents to adhere strictly to these guidelines. So that is why we had to perform a very strict comparisons of the characteristics of potential soil populations and of the recipient population, the population, to identify which was the best population to perform this, to transfer from which to translocate these nestlings. So the guidelines require, in fact, that individuals released into an existing population of conspecifics should originate from populations whose genetic constitution and behavioral traits are similar to those of the target population. That is, in this case, the target population is the population, and the recipients are potential other, and the source population are potential other populations in Italy, for example, or in the Mediterranean area. according to these guidelines, we devised two preparatory actions, actions A31 and action A32 in Lifefico. The first one was aimed at assessing the population genomics of the population and of the potential source population for hacking, while action A32 was dealing with the analysis of migratory routes of the population and to compare this with the other potential source population for hacking activities. Migratory routes and on-breeding range, of course, is also important. So I will briefly talk about action A31 and then let the floor to Jennifer for talking about action A32. So for the population genomics, we aimed at performing a genomic analysis of the population and compare it with other populations from the breeding range of the species. So thanks to very active collaborations by kestrel researchers throughout the world, we collected samples from the entire range of the species from Mongolia, from the easternmost populations in Mongolia to the westernmost population in Spain and Portugal. And overall, we performed RAD sequencing of 93 individuals, which RAD sequencing is a special technique of genomic analysis, which kind of identifies single nucleotide polymorphism, so polymorphism of single bases throughout the genome of the individual. And this was done for 93 individuals overall, 10 from which from the poplane, and the others from the 15 populations scattered through the entire breeding range of the species. On the whole, we identified 6,000 neutral SNPs, which were used for all the analysis of genetic population structure and for identifying the similarity, for analyzing the similarity between the poplane populations and potential other source populations. Here is a map of the sample population, as you see from in a color gradient from red to the west and to blue to the east. First of all, the analysis of SNPs and of RAD sequencing data provided a clear evidence for isolation by distance model of genetic differentiations. That is to say, more distant populations are more geographically distant populations are more genetically dissimilar, different than nearest one. So there is a kind of a gradient of genetic differentiations with distance. It is quite clear, and the mantle test provides us clear significance for this isolation by distance model. So this is the first basic results of the population structure of the species. When looking in more detail at this clusters of individuals by the population structure analysis, we clearly see that there are two main clusters of two main genetic clusters in the kestrels. One is pertains the easternmost populations, all the cluster altogether. So the Kazakhstan, Russian, and Mongolia birds, they all cluster together, and it's clear from the blue part of this graph here. While all the others, basically all the European populations, they kind of cluster together. And if you go with a K3, which is a bit more, let's say, detailed analysis, we also identified a cluster for the Iberian Peninsula, but it's not very differentiated from the one of the rest of Europe. So basically, we have two main clusters of population. So the K2 is the best supported population structure model, and it basically identifies and distincts very well the easternmost populations from the westernmost ones, from European ones. And yeah, and then it comes the comparison of the genetic similarity between the different populations we studied. And you can see this heat map here with also a cladogram on the right. And here, I will not go into the details of this complex heat map, but what you can see here is that the population labeled which Italy South and Italy North cluster very close together, and they are part of the same clade, as you can see here from this cladogram, and different, for example, from Sicilian ones. So from here, we identify that probably the Northern Italy population derives from the Southern Italy ones, and they are very closely related to, and the Northern Italy birds are very closely related to the Southern Italy ones, and different from the Sicily ones. So that means that the potential source population is identified as the Southern Italian population, probably. for performing hacking activities. So the best individuals for hacking should be those from Southern Italy. That is why we focus on Matera. But then, of course, we need also the proof that the behavioral there was behavioral similarity, for example, in migration between these potential source populations and the poplain ones. And I will leave the floor to Jennifer to talk about the migration part. Thank you.
Thank you. So yes, now the behavioral part. So within the Lifefico project, to investigate these behavioral differences between Northern Italy, Southern Italy, and other potential source population, we deployed GPS on the back of lesser kestrels, and in fact 53 GPS tags, and from which we retrieved 28 tags with data. So this is the tag that you can see on the back of the bird. And we deployed them in Matera and Greece. But we also had access to previous data from other Southern Italy populations, so Sicily and Apulia. So we could compare all these data. So what you can see in this map is the post-breeding migration, so that occurs around September. And you can see that the individuals from the poplain in yellow have a quite similar migratory route compared to the individuals from Sicily, Apulia, and Matera in particular. But they are more west compared to the route taken by individuals from Greece. Now, when we look at the pre-breeding migration, which occurs so when they arrive around February, March, you can see again the same, that individuals from Greece migrate more east, and individuals from Italy, south and north, migrate more west. And regarding the wintering behavior, or the wintering range, sorry, you can see, so again in yellow, the individuals from poplain have a very similar non-breeding range to the individuals in Sicily, but there were very, very few birds, actually only 4 birds tagged in Sicily. But they're also really close to the breeding range, the non-breeding range of birds from Matera and Apulia. And again, they are more distant to the non-breeding range of individuals from Lemnos. So these results of the migration and the genetics part are concordant and leads to the results that, yes, individuals from Southern Italy, and in particular from the Matera population, which is a large and healthy population, are the most appropriate ones to conduct the hacking activities. So in the next week, actually, we will start the first translocation event of the 30 first chicks that will be translocated to the nesting tower in the poplain. And we will continue to do that in the coming years to improve the colonization probability.
Okay. Thank you, Jennifer. Thank you, Diego.